Right, we're in the home stretch now. So the last part of our second line of defense is to discuss antimicrobial products, some chemicals that are around that assist in this innate immune response. So let's see, we've got interferon. So one way that an infected host kind of can combat a viral infection is with a family of cytokines that are called interferons. Uh, so interferons are a class of antiviral proteins that are produced by lymphocytes and macrophages, and they're typically produced after the body has come in contact with a virus. And one of the functions of interferons are to interfere with viral multiplication, right? Because it's kind of hard to affect viruses sometimes because they go inside a host cell. So this is kind of one type of chemical that we have that in the effects of a virus. So an interesting feature about interferons is that they are host cell specific. They're not virus specific. So interferons that are produced by human cells basically protect human cells, but produce very little antiviral activity for cells of other species, such as chickens or mice or things like that. So they're only good within the body in which they're produced in. So, but the interferons that are produced, they're active against a number of different viruses. So they're different interferons for different viruses. It's different interferon organisms, host organisms that are. So just as animal species produce different interferons, that individual can produce a wide variety of different interferons. So human interferons are of kind of three principal types. We have alpha, beta, and gamma. And each of the three types of interferon can have slightly different effects on the body. So gamma interferon is produced by lymphocytes, and it basically induces neutrophils and macrophages to kill bacteria. So the gamma interferon kind of kills those types of leukocytes and sets them into action, whereas alpha and beta interferon are produced by virus-infected host cells and produced in small quantities, and they actually diffuse out of the cell and detected by neighboring cells that haven't been infected by a virus yet. What they do is they cause that neighbor cell to then start to manufacture messenger RNA, which is that messenger RNA produces antiviral proteins. And these proteins are usually enzymes that disrupt viral multiplication. So it's kind of an interesting way of going about something. So here we see one host cell, and we've got our virus with our unique geometric capsid there. And so this virus is infecting this cell. It uncoats. We see the viral nucleic acids. It's starting to assemble more viruses, which will then be released and spread to other cells. So this infected cell, what it does is it starts to produce interferon. Interferon is secreted by the cell and it's detected by the neighboring cell. The cell, once it kind of gets wind of the interferon, will then start to produce antiviral proteins so that when it is affected by that virus, it can quickly retaliate. And those enzymes will go in and of the virus. So it's almost like an altruistic response by the one cell, if you think about it. He's kind of like, hey, I'm affected, save yourself. To neighboring cells that these viruses are around and you better arm yourself and get ready for infection. So besides interferon, we also have what's called the complement system. So this is a defensive system that consists of over 30 different proteins, and are found circulating in the blood and in your interstitial tissues. They're in every fluid in your body all the time. So these proteins are around all the time. Not like they're activated or they're marshaled up after an infection. They're already present. So the complement system is named because it complements the cells of the immune system. So it's going to help in destroying pathogens. It's part of our innate immune system because it doesn't change over time, but it also can assist in our adaptive response as well. So proteins of the complement basically destroy microbes, mostly by lysing the cells. They can assist in inflammation, the inflammation process, and in phagocytosis as well. So typically complement proteins are inactive until they're split into fragments. And it kind of creates this cascade effect. So here we've got one molecule of the complement system. So it says C3. So when C3 is lysed, kind of broken down into what we call 3A, C3A and C3B, then those two products, they become, the fragments are basically activated proteins, and they're indicated by having those lowercase letters of A and B. So typically in complement, the proteins are numbered C1 through C9, and the numbers just simply indicate the order in which they were discovered. And then you get the A and the B's are used to name the fragments that have been activated. So we've got the inactive complement protein of C3, and then once it splits, we've got the A and B fragments which have been activated. So the activated fragments kind of carry out the destructive actions of complement. But it's like a whole cascade of events because that 3A and then 3B go on to bind to this C5, which causes that to split and become activated into A and B. So in other words, one reaction triggers another, which triggers another, and then more product is formed. So with each succeeding reaction, the effect gets amplified many more times as the reaction continues. So, sorry, I'm trying to find something. I think I have it in here. All right. So what you're seeing at the bottom here is the membrane attack, or the MAC. So the fragments bind together, and so all these fragments that have been activated, so they'll bind together. That's why we kind of have this little rainbow molecule here. And they insert themselves into the plasma membrane, and they're going to form a channel. And so you've got this channel that's now inserted into the membrane, and this is called the membrane attack complex. So the membrane attack complex. And this creates a channel or a hole, and that's going to result in the lysis of the cell because things are going to be able to leak out. And that's what they're showing you in this electron micrograph image. The holes that have been created by the membrane attack complex. So you can get many of them over the surface here, and this is going to cause the cell to lyse. So that's one way in which the complement system helps in the immune response. Other fragments can cause increased blood vessel permeability, which is then going to assist with inflammation. Okay, so there's a variety of different things that they can do. So now I'm going to go back. There we go. So there's a couple of different complement pathways. We've got the classical complement pathway and the alternative complement pathway. The classical pathway was the one that was first discovered, and this pathway is initiated when antibodies bond to the antigens. Basically, they bond to the pathogen or the microbes. And the alternative pathway does not involve any type of antibodies produced. This pathway is activated by contact between complement proteins and the actual pathogen. Where we've got our I want to say MAC attack, or membrane attack complex, that creates the holes in the wall there. All right, lastly, we have a more recent discovery of what are called antimicrobial peptides. So proteins are made of polypeptide chains, right? And polypeptides are simply amino acids that are strung together. Bonds. So in this case, we're looking at kind of there are 12 to 50 amino acids that are produced by the ribosomes. So these are very short peptide chains that we call antimicrobial peptides, or AMP. So even though these are kind of fairly recently discovered, they're considered still to be a very important component of our immune system. They were first discovered in the skin of frogs and in some insects, and then later on humans in neutrophil cells. There's, lots of different types of AMPs. They've found over 100 of them. That's across plants and animals that have these. And these peptides are active against bacteria. They work against viruses. They work against fungi, even eukaryotic parasites. So they're very effective. Basically, the cells produce AMPs when microbes attach to the purr, so those receptors on the outside. So AMPs can be produced by the cell in response to adhesion of a pathogen. Mode of the AMP varies depending upon the type. So it could be that it inhibits cell wall synthesis in the pathogen. It could form pores in the plasma membrane, like we saw with the complement system. It could destroy DNA and RNA. So there's a wide variety of different types. Defensins are AMPs that are produced by neutrophils. There's ones that are called thrombocytins, which are made by platelets. These AMPs tend to be stable across a wide range of pH. They seem to work kind of flawlessly with other components of the immune system. So, and best of all, pathogens don't seem to have shown any kind of resistance to AMPs. So there could be potential therapeutics value to these. So this image here, it comes from your book, and it's showing AMP in purple. So usually proteins are shown in purple. So it's showing a very short polypeptide chain that's folded into a certain shape, squeezing in between phospholipids that are part of the membrane. Because it's got this kind of big shape,